So now we have time for all kinds of questions and discussions. So anyone who has any, feel free to either blurt it out or just raise your hand through Zoom. I'm not seeing anyone. Okay, so yeah, Bob? You're muted. There you go.
Yeah, I wanted to ask John Polymani about his compliance of microvessels. And the issue here being whether they are whether they're really elastic or whether they're actually quite flaccid, but their failure to expand very much is simply to do with the fluid outside them. And the vital point which actually allows the capillary microvessels to do their job is that the capillary microvessels are actually very porous.
Yes.
And most of the water that goes in at one end of a capillary doesn't come out immediately, it goes into the tissue and then comes out later. So as that's what oxygen 15 PET depends upon. That suggests that when a capillary expands, it's a transfer of water between its inside and its outside rather than a real expansion against a tension in the wall. So I'm wondering, is that built into the Van modeling?
A little bit, actually. So thanks for the question, Bob. So we read with great interest your paper, 2012, Krieger, I believe is the first author. Yes, and that had a lot of insights into the potential role of water extravasation, in kind of managing capillary compliance. And so all I can say is that we have looked into this. So Jorg, who's here, he has implemented a little bit of water permeability, both in order to try to understand, non-bold contrasts, but also to understand the influence of this sort of release and pressure on not only the capillary compliance, but maybe also the downstream compliance of the veins. If there's some pressure release in the capillary bed, as you say, maybe that leads to less pressure delivery to the downstream veins. So all I can say is that we've looked at it. It seems that it's having a small effect on the overall picture, but there's other factors at play. I think another lesson from that work is also about how does intracranial pressure change, with activation, and could this have some effect on compliance as well? So all I can say is that we've looked into it. It seems that so far, incorporating this water extravasation in terms of understanding pressures is playing a kind of minor role, not changing things too much. But I think your point's well taken, which is that
I don't think we ever expected it to be sort of a huge mechanism, but the point was that there needed to be some way of understanding how capillaries could actually. The other issue there, and this is the second point really that I wanted to raise, is how much of David Atwell's lab's work on the distribution of pericytes is going into this as well, because of course the spatial specificity of changes in blood volume depends enormously on exactly where the pericytes actually are.
No, absolutely. We're starting to get some data on this. We're starting to get some data on the distributions of pericytes and smooth muscle cells in the human cortex to try to get a sense for where these control structures may be. On the flip side, as you know, we've been looking at work from Andy Shee's group a lot. The role of pericytes in functional hyperemia, is still a little bit questionable. As you know, probably better than me, there's some debate about whether these pericytes that are shown to be active are really on capillaries or precapillary arterioles. It seems like Andy Shee's lab is showing that pericytes may be involved in kind of longer scale kind of adjustments of flow and almost like a longer term kind of remodeling, kind of fine tuning of flow over time. So I guess all I could say is that we've been looking at this a lot. Still seems very controversial. We also know that, gosh, these pericytes, as you know, are so sparse once you look in the capillary bed. And they really don't have the ability, it seems, to really regulate diameter. Their processes don't really enwrap, the capillaries. And so it's still unclear to us. I mean, the point is all we can do is look at the data and try to understand what it's telling us. But the bottom line is today, I think the role of pericytes in terms of functional hyperemia on the capillaries is still a bit unclear. So all we can do is model both, which is what we've done here, and look at the differences. And so I guess
I'm really impressed by your modeling, I have to say. It's very nice to have something you can actually stick these different parameters into. Yeah,
exactly. It's this little laboratory. We can say, okay, what if pericytes do dilate? What if the capillaries? What if they don't? How does this affect the signal? But I guess the only other point I would raise is that, Mike and Nadegaard is also showing, that, in terms of fine scale regulation, that the red blood cell deformability, also may change as oxygen is released from the RBCs. And this can also be another, it's pretty cool. She's got some really nice data showing that maybe, blood viscosity changes with activation, so yet another, complication in our models, but the point is that, jury's still out. I mean, we're trying to at least get some statistics on the distribution of pericytes in human tissue. It's, so far been tough. But something that's definitely on our radar, and we're reading these new papers with great interest to try to understand their role.
Thanks a lot, John.
Yeah, thanks, Bob.
Nice to hear from you.
It's nice to see you.
Yeah.
Are there other questions?
Just maybe an obvious question, but obviously, John, Amir, and maybe even Evelyn can all pitch in on this. Obviously, John, you made the point, like, the contribution of different veins depend on their orientation to be zero and stuff like that, right? And obviously, Amir pointed out the need for a higher resolution, and right now we have mostly one voxel per layer. But even if we go higher resolution, one of the main approaches that we do in layer-fMRI, especially from the fMRI perspective, regardless of the sequence that we're using, regardless of how we are acquiring our data, tend to average all the voxels within a layer. Sure, we can split within layer, across layer, that's a completely different story. But once we do that, because of the complexity of the fold of the cortex, obviously it does not downplay the importance of understanding the biophysical origin of the signals, but how much of an impact will be to be accurate in modeling the signal, taking into account the fold of the cortex if we average within each layer? See?
Well, maybe what I would do to try to address that question is maybe expand upon it, and that is, how do we apply these models to our data? I mean, how do we take, like a van, for example, that was made from a mouse and use it to interpret, our 70 human fMRI data? There's a lot of work that has to be done, I think, to build that bridge. But no, you're absolutely right. I mean, gosh, one of the reasons that we're also careful about distinguishing between kind of cortical depth dependent signals and laminar signals is because of this complex relationship between the layers and the folding pattern. We've characterized this a lot in terms of the, histological layers, less so about the vascular layers. But the point is, it's well taken. And so I think, trying to understand, this kind of gets to Tyler's question too, generalizability. How is it that we can take these models? When are these models generalizable enough, that we think that we can apply them, to our human data? And that's a big question. So you're raising one important challenge. I think there's others as well. Maybe I'll let Amir chime in, but the point is, definitely something that we need to take into account. Understanding the relationship between the folding pattern and the layers also, doesn't seem to be constant across the brain. There's not a simple rule, and so lots of complexity there. Amir, did you want to chime in?
Yeah, so I would just say that, for me personally, laminar fMRI and understanding laminar fMRI is the most challenging question that I've been looking at throughout my career. And I agree with what John said, and I think that we need to improve as much as possible spatial resolution, signal-to-noise ratio, a gradient system, RF coils, pulse sequences, etc. But at the end, as long as we use blood in any kind, we will be limited to what we can do just with fMRI. And so we will have to incorporate information in a hierarchical manner, like from starting with mice, going to rats, going to monkeys, and whatever we can do in humans in order to create meaningful models with assumptions. Now, this is true for anyone who wants not just to show, okay, I was able to demonstrate selectivity at a certain depth to this stimulus or the other, but really to understand how the brain works. And I think that everybody in this meeting understands that a lamina resolved function is extremely important to understanding how brain works. What we bring to the table is being able to do something along these lines in humans, but it has to be connected to a lot of groundwork in animals on the mechanism.
That's a really nice point. Evelyn, do you have anything to add for that?
I would say I'd just chime in on one small aspect of what Amir was saying. I completely agree, but I would try and put a positive spin on it in terms of there are many advantages to having bold or a vascular measure that's complementary to the neuronal or electrophysiological, or in my case, optical images that we can acquire that have more specific cellular origins to their signal, with all the caveats that go with those statements, because you do have breakdown in neurovascular coupling in many diseases that are my personal interest of looking at. So when you have two measurements, you get to see a little bit more about what's going on when something goes awry. So I wouldn't say we have to be quite as humble about, oh, we just look at bold or vascular signals. We can also be a little bit proud of that. That being said, I've been arguing with neuroscientists for the last 2 years, but there we go.
Perfect. So Federico, you had your hand raised.
Yeah, I have just a question for everybody, just a panel. I think it's super interesting to see how we are finally thinking about using biophysical models from different levels, right? So neuronal biophysical models, Amir showed some vascular biophysical models connected to actual physical models of the signal. And to me, it seems like at the moment we are in this expansion phase. We are trying to make the models more complicated, right? So because we are thinking, everybody's thinking, oh, we forgot this, oh, we forgot that, we should, oh, this is important. But from my limited understanding of modeling, I know that one important part of modeling is actually then going to be model simplification, right? So we'll have to then boil down to what is a model that is meaningful for us. And I wonder whether we have ideas in our head of how we are going to do this. Because if we are going to do it only based on fMRI, we may actually end up with a model that is not meaningful, or maybe only partially meaningful. So I think that what Evelyn showed is inspiring because it does provide us with tools. But I also think, like, how are we going to go there, right? So how are we going to decide what is relevant for us in human neuroscience, for example, my field, and what is the minimum model that we could play with? It's quite challenging. I don't have an answer. I don't know if anybody has an answer, but I'm scared, right? Because in the last years, I've seen models growing, and Amir just showed us a neuronal model that is even more complicated than the models that many of us were thinking we were going to use. So I'm like, okay, so now the number of parameters is exploding. So do you have any thoughts? I mean, what kind of thoughts experiments you would do?
Can
So,
Bob as a thought, so maybe Bob can answer my question.
I just want to quickly chime in on this. I don't want to say much more about this, but I just want to remind people of Bob Schulman's really powerful statement that what really matters is energy. And if one can get the modeling down to simplified enough that we're just talking about connectivity and energy, just how much energy is being consumed, never mind how many action potentials per second or or what is the calcium level or anything else. The bottom line is actually energy, according to Bob Shulman. And I just want to remind people of that, because I think that's kind of forgotten recently as a real marker of what's important. I'll say no more about it for now.
So I would like to comment on Federico's question. Well, first, you know, the model that Sotero and I published is indeed complicated, but it does the work. If you look at the paper, it reproduces, and we showed objective evidence that it reproduces the connections. Indeed, it is complicated, etc., but it does the work. My second comment on that is that I think that a hierarchical approach to modeling is important. And I believe that that's what the European Human Brain Project does. You know, they started big, but already at the start, they said that it will be hierarchical, meaning at any point, you know, they will take a small part of the problem and will try to model it and to reduce the dimension. The last comment on that I want to make is that I'm very much in favor of probabilistic models instead of you know, models that give one answer. They can manage a very large number of dimensions, many variables. One, I believe, a nice example is the paper of Chaimov, or how is it called in the US, Chaimov, in Neuroimage 2018 about the point spread function. Here is Dennis, hi Dennis, about the point spread function of bold response. So, you know, like we had really a large number of dimensions, but by incorporating a probabilistic model and by searching efficiently the space of the possibilities, I believe that we gave very good results that again, we validate. So, you know, I'm not worried about too many variables. There are mathematical models now to approach them. Of course, we don't need to exaggerate by that. And I'm very much for probabilistic models, and I'm very much for hierarchical modeling, solving one problem at a time.
One quick comment as well. I really enjoyed all the talks, and certainly John's modeling was it was super impressive. And I just want to add one thought about modeling. I mean, you know, obviously, you know, it's always good to try to you know, model to get some sort of insight as to potentially what's important or not. You know, and certainly there's insight provided today. But certainly also, you know, I think the next step, as Bob was sort of suggesting as well, is that, you know, even with looking at energy, for instance, I mean, and the goal of the model is actually to try to figure out what's important, what's not important. And also, but towards the goal though, I mean, you have to, obviously, if you have models, you can maybe calibrate better or you know, try to find ways. I mean, there's two approaches: one, calibration, or being ever more selective with your pulse sequences, or maybe a little bit of both. You could look at the ratio of spin echo to gradient echo, or you could look at the ratio of flow to volume, or, you know, and to try to get at, because really, I mean, this varies. All these models, all these parameters might vary on a voxel-wise basis in some way, and so you have to sort of make these maps that you could potentially calibrate. And I think John is going in that direction in that regard. And maybe you can get calibration to get, you know, obviously early on, you know, you can extract CMRO2, which might be, you know, obviously this is very noisy, but that might be the best measure of looking at layer-fMRI. So I think we're making progress in that regard. And I think everyone knows that, yeah, I mean, these models are great, but you really have to get a calibration or a more specificity with your pulse sequence.
Well, of course, we have to do calibration, right? The model is just a model without calibration. It needs to be fitted to measurements. And the more measurements we have, and the more multimodal measurements we have, the better we can specify the model and be sure or be reasonably sure that it's going in the right direction.
So all I would add, just to kind of, I agree with Federico. We need to also, because of the complexity, these models can really start to explode. And as we try to introduce more and more realism, we introduce more and more parameters, and there's a real danger for overfitting, as I said. So as simple as possible, but not simpler, right? And so I guess the question is also, what do we want to do with these models? On the one hand, the balloon model is awesome. It fits so much data. If you want to look for activation to a GLM, the balloon model is great. But gosh, it's the parameter values are a little bit tough to interpret, right? And so if we want to go further, if we want to understand more about these signals, I do think we do need to embed more realism in some of these models. But I agree with you, Federico. I don't know how to strike that balance, right? I mean, we have to be honest about the complexity, and we need to be careful about, trying to embed more and more realism with kind of diminishing returns, right? But I'm not sure how to strike that balance. All I can say is that you're right, that we need to push to make them as simple as possible. But we have to ask, what are we using them for? Are we using them to, interpolate data? Are we using them to identify activation? Or are we using them to try to interpret? To try to, as Peter said, try to calibrate the data, to try to extract, more information about metabolism or energy, like from the data. So yeah, so I think that there's no one answer, right? I think that the balance gets struck differently for different questions. But the point is we need to be thoughtful of how to strike this balance.
I wonder, if I can just say, I wonder if it's not time also to, or there will be a time to, I'm thinking like, I like what you have done, which is this idea of being able to play with all the parameters. But if we were able to all do it, right? So suppose you have like a web application where everybody can play with a toy model, and we can all kind of do some kind of sensitivity analysis on a parameter such that, for example, for a thought experiment that we have in our head, I go ahead and check whether a certain parameter of the model actually matters. And then I leave you a note and say, look, I've done this, this, and that, and I change this parameter in this range, but nothing changes in the output. So maybe we can get rid of it, right?
Yeah.
Maybe it's a way to speed up things, right? Because it's a little bit, it's a lot of stuff. And I know this is what has been done in the neuronal modeling world, right? Where you will go from neuron models, right? Really neuron models to mass models. And that's what we want to somewhat achieve. I think we need to get together because there are too many parameters for the people that are in this audience already. So somewhat, I think we all have enough work if we were just to take one of these models and work for the next 10 years in working with one of the parameters each. So maybe is a thought-provoking idea, right? The one of saying, why don't we just crowdsource this kind of work, right? So it is a community. We could just do it together, right?
It's a great idea. I mean, yeah, I think that, for the models that we're playing with, we do need to make them a little bit more practical. Again, that's another motivation for making simpler models, is just that we can't have models that take 4 days to run. So yeah, working on that web app, but it might take some time. But no, you're right. You're absolutely right. And I love this idea of model selection and getting rid of parameters. But it's also true that as Bob was saying before, when we first were looking at water extravasation, like, is this a factor? And it's good that we tried it because we weren't sure, and we implemented it, and it had a small difference, and so right now we're not including it. And so that was one example of what we needed to try. We needed to do this sensitivity, as you said, to see if this one parameter did make a difference. And once now we have confidence. Okay, we don't this is an important feature. It might not be relevant for certain aspects of the model, so we're able to kind of exclude that. And so maybe just more of this. More of this, testing parameters, as you say, do the parameter sensitivity and simplify.
I'm wondering if if the data that Evelyn is collecting, where it's very multimodal and across very different aspects of what we're recording, is capable of stepping in to do a little bit of that sensitivity analysis for us.
Sure. Ideally, yes. That's something that we're trying to work on, and actually we're anxious to try and collaborate with folks who have much more modeling experience than we do. So if there is interest there to reach out and share some data and play with some of these parameters, there's definitely interest on our end. And not to make things more complicated, but it also depends on the population you want to apply your model in. So maybe we have more than one model that's well suited to, say, a neurodevelopmental population, another model that's well suited to, say, someone who has very hard veins, for example. So it's one size is not going to fit all, and maybe a little triage at the beginning is going to help sort out some of this as well.
I think we have a few people lined up for questions. Avery, you had your hand up, or was it answered? Perfect.
No, sure. It's a bit off the topic of modeling, I think, but it's still relevant to the discussion. So thanks so much for all the speakers today, too, and for the organizers for this. Quick comment to Evelyn, I guess, too. You know, we're putting together this layer-fMRI analysis survey because we feel like in the layer-fMRI world that, you know, there's only, you know, maybe on the order of 100 centers doing high resolution fMRI. And so we kind of need to band together and find kind of the best ways to do our data analyses. Whereas I think we have to be even more sympathetic to you, where you're maybe like a handful of centers doing your sort of data analyses. So lots of challenges there, I'm sure. And it looks like you've got a great suite of tools. But I had a question for Amir. You had mentioned kind of these two contrasting cases of stimuli where you had these kind of very rapid, brief stimuli where you saw very brief evoked neuronal activity. And then you also contrasted that with these long duration, like 20-second flickering checkerboard sort of stimuli. And you said, okay, in one case, you know, we have such brief neural stimuli, we can't do fMRI. And in the other case, we have these, you know, kind of pronounced typical bold responses, so we can do fMRI. I was wondering if you can maybe sort of unpack that a little bit. Like, is there something in between, you know, where is this transition between we can't do it and we can do it? Or in the case where you don't have such a regular interstimulus interval with these very brief stimuli, do you think that maybe you can start to see you know, hemodynamic response, even if it is a really brief stimulus? Yeah, just kind of wondering what your thoughts are on where's the middle ground where we start to, where things really do kind of become feasible or infeasible.
Yeah. Do you guys see my monitor?
Yep.
Yes.
Okay, great. So thank you for asking. Indeed, there is what to unpack. So, you know, what I just mentioned briefly during the talk is that if we use stimuli that produce transient responses, then they are diluted. The responses that we get are actually diluted with fMRI in response to spontaneous activity. But there is more to it because these brief responses include inhibition. So there is a lot of inhibition over there. There is a paper of Seiji Ogawa from approximately 2000 showing that he gives electrical responses at a brief interval. You know, he looked at how I produce a time course of neurophysiology from that. But really what he did is the bold response reflected the inhibition. When you get the stimuli, transient stimuli that cause transient stimuli, when you get them close to each other, really inhibition is coming into play and it reduces the overall response. Now, I agree, we should find something in between. I can tell you that when I look at human fMRI, I'm looking at anywhere from 4 seconds to 12 seconds stimulation. You know, sometimes I do use baselines in between just to understand how the hemodynamic response changes. Maybe not as we used to do in a long time ago with block stimuli, but somewhere in between, I do that with human functional MRI.
So I do want to chime in a little bit here. This is something that we struggled with a lot because in the mesoscale optical imaging world, they'll do a very short stimulus, 1 second, done. And in fMRI, as Amir was just saying, you get very long stimulations, and you go to a calcium imager and say, we're going to stimulate for 10 seconds. And they're like, what are you going to do to the neurons? Honestly. So, because they will have a very nice big stimulus response, they'll go almost quiet, this excitatory neurons, calcium imaging. And then when you turn the stimulus off, they'll fire again. So you'll have this very strange profile in your calcium imaging data to try and reconcile it with your bold response. So we did spend a lot of time sort of debating, okay, how short can we go with bolds? And we did settle on about a 5-second stimulus and was able to get something that was reconcilable between the two data sets. But this is something we're actively thinking about in terms of what are the excitatory neurons doing, what is inhibition doing during that time, and what happens also at the stimulus off time point within both the data. So it's very happy to discuss further on this point.
Actually, sorry, very good follow-up to this question, because I'm quite interested in like stimulation time and stuff like that. When you say 5 seconds to reconcile different modalities, you mean 5 seconds at interval between the stimuli, or 5 second continuous stimulation?
5 seconds on time. So we do a very sparse stimulation. We only stimulate once per minute for 5 seconds.
Thank you.
But that's enough to have adaptation. So of course, I mean, that's what happens. But that's a thing that can even help you, right? Because in some way, because it's a stimulation paradigm that will allow you to both see excitation and local inhibition in some way. So of course you need to decide that goes to my point of like, what are the thoughts experiments, right? Because maybe brief stimulation is not the best way to challenge something, or continuous things will do other things, but we need to know, right? So what exactly we are going to do in order to fix these networks in some way, because it's not just that repeating a stimulus is better. Repeating a stimulus makes things different in cortex, right? So it's not the same thing for the brain. It may make it better for the fMRI, but it's not the same thing.
I totally agree. Yeah, I think we're more in agreement than you think. So even
I
if
agree with you. I mean, what I'm
Right.
saying is that I'm just saying it's not necessarily a bad thing, right? A variety of responses because of temporal effects, even if combined, can help you somehow. Even if in the end you average this thing in fMRI, maybe there is a way that is still reflected, right? Maybe a little bit of adaptation will reflect in some way in the overall bold compared to no adaptation at all, or a lot of adaptation, or so on and so forth, right? So parametrically modulating this could actually be a way to help you figure out what are the relevant things instead of just choosing one, right? So saying, what about instead parametrically modulating the length of the stimulus and just see what happens? I mean, I think this has been done millions of times for studying, for example, the hemodynamic bold response, right? So it's not a new idea. It's just that I think there is really a lot of good in variety, and in that, yes, in experimental variety, not in. So while model needs to be simplified, experiments need to become more complex so that the simple model fits to the many experimental variables, right? So that's somewhat what we are aiming for, right? I guess, right? So that's what we are all after.
I think I'd make one point based on what you're describing a little bit is like, I mean, you have even more subtle complexity than that. In every individual, even if you present exactly the same stimulus, which we've done with our multimodal experiments, you get different varying responses. Sometimes you have a bigger bold response and also a correspondingly larger calcium response to the exact same stimulus. And others, like especially 2-photon microscopists, will tell you time and time again that they can do exactly the same thing as far as they're concerned, and they see something different. However, as we move from the microscale to mesoscale to macroscale, we do have sort of a summarizing effect. Like every time you see a I don't know, your cell phone, you have somewhat of a similar response in the very end. There's some sort of summary that's happening down from the neuron all the way up to the actual human behavior. So maybe that can be a little bit of consolation in that even though we have this marked heterogeneity, when we're looking so closely at the data, there is also some sort of summarizing effect that happens as you go upscale.
Yeah, although maybe I don't want to console everybody too much, but there is a lot of variability in single trial responses in fMRI in humans, right? Even if you do it at 3 mm isotropic. Variability that is understudied completely, right? Of which we know behaviorally it's meaningful, right? So there are beautiful behavioral experiments that show that this variability is actually meaningful. But for decades by now, we tend to summarize. It's not that it's the response that is summarized, I think, it's that we summarize. And I think that's important to remember, right? It's not that the body is necessarily blind to these variabilities. It can be probably seeing it. It's just that we see it through the eyes of doing statistical tests with general linear models on average of trials, of hundreds of trials, and we just interpret the average. A very simple thing from a statistical point, we use random effects across subjects. We should do random effects across trials, right? So that's the obvious thing that one would have to do. It's very complicated, but it's the actual variability that we care about. More than the noise that we measure regularly in fMRI as the residuals, the actual variability that we care about is the trial by trial variability.
Absolutely, you're making my arguments for doing simultaneous imaging.
Yeah, absolutely. Yes, I agree. I mean, that's why I said it from the beginning. Your experiments are, or your toolkit is great. So.
Just to remind people for another blast from the past is the work of Ariely and Grunwald, where they use voltage-sensitive dyes, and they found that their results for a particular stimulus seemed incredibly variable. But the variability reduced tremendously when they used as their baseline the activity map just before the actual stimulus, and used that as and then they found that in fact it was extremely consistent. So that example has been hauled out a few times over the years, but I don't think it's ever been taken quite as seriously as it should be. It may be no coincidence that as Evelyn was saying, the net result may be on the macro scale actually the same, because what it really counts at the micro scale is actually the same. It's just that it's on this very variable baseline of all of the housekeeping and stuff that the brain is actually doing.
Yeah, I think it's related to what Amir showed, right? Also alpha modulating. So pre-stimulus alpha or low frequencies pre-stimulus are known, but there are, I also want to say there are also a lot of behavioral studies and also non-behavioral studies that show that there are indeed variabilities that are meaningful, right? For example, I don't know, if you just need to hear a very random noise once and then hear it again a second time, the response of the second time is different than the response of the first time. And that difference is meaningful because it shows a memory trace. And I mean, there are many studies like this and many similar things that show that variability at that level, of course, having taken away everything that we can explain, Bob, in this I agree with you, right? So we have to be also good in understanding that that variability will be partitioned in many parts, right? Some of it we will know, and some of it we will have to understand.
The real question is whether it's random variability or whether it's variability that's associated with a history.
Exactly. Or yeah, with a history or with another physiological or with something else like alpha power, pre-stimulus alpha power, or yeah.
So I want to add something that again may sound trivial, but I've been experiencing it recently, and I'm not sure how much we are aware of that. And this is subject sleeping in the scanner. It's unbelievable. I started doing now more functional MRI in humans, and I record video of the eyes. And for a long time, I knew that with monkeys, I need to wake them up from time to time. And believe me, it's worse with humans. I know Human Connectome Project did analysis with EEG, etc., and decided retroactively that a huge portion of the subjects slept. Let me tell you another something kind of funny. With this Yakub at University of Minnesota, at some point, we stopped seeing responses, and it turned out that the subject fell asleep. I recommend to everybody to record video of all the subjects. Look at the eyes. I think that this accounts for a large part of the variability. And of course, what Robert and Federico mentioned is correct as well.
No, absolutely, Amir. All I'll say is that when you ask subjects to fall asleep, they stay awake. When you ask them to stay awake, they fall asleep. And so that's one trick. But Katie Chang has some nice tools, actually. She's been able to use these EEG data, to try to extract patterns from the fMRI signal that do seem to be kind of evoked during sleep. And gosh, I mean, in our data, the bold signal just goes wild when people fall asleep. So there are certain signatures. I mean, we used to use EEG, but we can almost stage now by looking at the bold fluctuations. So that's good. That's good news. At least we can do some quality control. But no, it's a very important point. I think a lot of these data, we know that the networks change a lot during sleep. And so a lot of these data, may be corrupted.
I think there was a question from the audience. Mario, are you still here?
Yeah,
yep.
Thanks. Yeah, first of all, thanks to all the speakers and the organizers for the nice conversation. I've enjoyed it very much. I totally agree with the idea that we actually need to simplify the models as much as possible in order to guarantee or make easy the application of the models in a wide manner, and maybe eventually also in the clinics. However, I'm a bit more interested on right now on looking at new parameters. For example, I think that a way to also maybe simplify it or help simplify the model is to look into parameters that we can already measure with multiparametric neuroimaging approaches. So in that regard, for example, looking into the capillary transit time heterogeneity is something that we can already measure with multiparametric imaging. And therefore, I will also be maybe interested on hearing John's opinions on the possibility of how this transit type heterogeneity can actually influence the tissue response at the different layers, for example, because it has been shown that already like it has a global effect, like for example in pathologies like that alters flow, like for example internal carotid stenosis, that can also have also maybe an effect at the laminar level. Have you maybe thought into looking at it?
Well, a little bit from the other perspective. We were actually it was funny. We were just looking at Lee Fostergaard's work just a couple of weeks ago to think about, to what extent could we try to quantify this, right? So instead of asking, does transit time heterogeneity or the reduction of transit time heterogeneity during activation have an effect, we were trying to measure it, right? So if we do turn on the arterial flow, like how much does the transit time heterogeneity decrease? And can we try to quantify this, because that would be an excellent prediction that we can test against to measure data. And so I guess all I can say is that we've been thinking about it. Now the question of how does the capillary transit time heterogeneity affect the responses? I mean, I guess I view it more as the result of the responses rather than, kind of a driving factor, right? So the idea being that when the precapillary arterioles dilate, we do expect, some decrease in that transit time heterogeneity across the capillary bed. And so, and again, we know that there's some evidence that this could potentially increase the efficiency of oxygen extraction into the tissue. This is, Leaf's theory. So I guess what I would say is that we've been thinking about it. We've been trying to quantify in our models, how much does the transit time heterogeneity decrease? Now your question about, we do know that the capillary bed is really complex. Sylvia Lartois has been looking at this a lot, right? We know that The capillary bed isn't 6 kind of independent regions. As I mentioned, there's a lot of trans laminar flow. We know that there's a lot of shunting. We know that the oxygen of the red blood cells doesn't just sort of decrease monotonically from the arterial side to the venous side. It's much more complicated than this. Other complications, looking at distributions of hematocrit, to see if this might affect. I'm thinking about Federico's comments too. It's like, gosh, there's a lot of details that we could embed in these models. So I guess the short answer to your question is that we've been thinking about it. We've been trying to test, see if we could use these models to test these theories. There's some nice work by Andreas Linninger, who was looking, I think, at some of these effects, right? And I think Bruno Weber is looking at some of these effects as well. But all I can say is that we're trying to measure this. Now, how this could impact oxygen delivery across layers is a great question, right? I mean, if some layers have less of a change in capillary transit time heterogeneity, then this might give us some insights into metabolism in those layers. One thing that I haven't talked about is how do we model, a metabolism? In order to, get bulk signal, we need the tissue to consume oxygen, right? So I left that part out. Yet another piece of complication that we want to remove. But I think you're raising a really good point, and that is there's lots of ways in which the vascular architecture, doesn't just impact flow patterns, but it can also impact oxygen delivery to the tissue. And yeah, we should again, we've been thinking about kind of modeling this to quantify it. I guess that's the best way I can answer your question.
Thanks. I mean, it just basically throwing more wood into the fire.
Well, but it's a great point, right? Because what we want are, predictions that we can kind of confirm experimentally, right? So if we could measure this, and then compare it against our predictions, that'd be awesome. Sorry, yeah, Bob, did you want to chime in?
Yeah, I just wanted to comment on this particular point, that just to remember that the capillary vascular architecture is actually bootstrapped, because the capillaries grow in response to oxygen demand. So the pattern of capillaries that you actually see is somehow an expression of what the neurons are actually needing. And so that's a kind of constraint. It should be a constraint on how they're actually modeled.
Absolutely, and it's a confound as well, I guess, right? I mean, we know that, if the capillary density is highest, exactly in those regions that are most active and the layers that are most active, then yeah, that makes our work more challenging. But no, you're absolutely right, Bob. I mean, and that's another reason why we're scared about variability across individuals, right? I mean, if everybody has sort of a different angioarchitecture, then it's gonna be tough, to the extent that this impacts the modeling. And so, yeah, what we're trying to do now, as I mentioned, just try to quantify how much this capillary angioarchitecture varies across individuals, just to get a sense for what we're dealing with. If it's, on the one hand, if it's quite variable, that's interesting, it tells us that, the capillaries are another measure of, plasticity or modeling. That's meaningful. It's something that we should, that could be, important to understand, metabolic needs and, neural plasticity as well. But if it's more, uniform across individuals, then at least that makes our modeling efforts, more straightforward. But that was an excellent point. Yeah.
One of the beauties of this is it's actually available to MRI, vascular density, if we try hard enough at a layer-dependent level too.
No, absolutely. I think most of the methods, contrast in agent-based methods, vessel size imaging, capillary density imaging, the Kislev and Tropez models, I think are great, or good old-fashioned CBV is also a pretty good proxy. No, you're right. These are things that we can at least measure in humans. And so we don't need to necessarily go to animal models. No, that's a great point.
John, sorry if I maybe interrupt others, but have you thought about, well, I think, of course, if you can measure experimentally, that's the nail, right? So that's how you do it. But, the other old style way is to just understand how much do you have to vary it in order it to matter?
Yeah, exactly. What you were saying before. Exactly.
I mean, why that seems to me something that is still viable as a solution, because it is a thing that maybe you realize that you would have to vary way beyond what any kind of physical limit is for it to actually matter, right?
No, you're absolutely right. And so these models now that we're able to kind of parametrically vary, for example, capillary density is awesome, right? Because now we can vary it a lot, and if it makes no difference, one less thing to worry about. You're absolutely correct. I mean, it's true too that, honestly, because there's so little data on how much the capillary density does vary across individuals, we thought we should at least, try to make some measurements to kind of at least bracket, what the range is. But you're absolutely right. We should test it, do a sensitivity measure. If it's not relevant, we have so many sources of noise, we can just remove it and try to kind of pare down these models.
It's a long process, obviously, but of course it's the progress that we have to make. So this is a very, very interesting conversation, but I'm also a little bit aware of the time and that other people probably have other meetings that they'll have to jump into. If there are any really absolutely burning questions, we can discuss them briefly, but otherwise, Luca, do you want to say anything to wrap up?
No, thank you so much. Thank you to the speakers and obviously to the people that came to listen to talks and generate the discussions. And no, that was like a great meeting. As we all can agree, we really enjoyed all these stimulating the conversations and talks about some very important points. We need to understand where the signal is coming from, right? That's ultimately, that's been the challenge for fMRI for a long time. And now that we're going to do laminar fMRI even more so. So no, thank you very much to the speakers and to everyone else. And stay tuned. Check out the website, interact with us on Slack, and we'll have another meeting soon, hopefully.
Thank you so much.
Thank you.
Thanks, everybody. Thanks for organizing this, Luca, Tyler, Renzo. It was great. Thanks a lot. Take care, guys.
Thank you.
Hope to see everybody soon.
Hopefully see you in person soon.
All right, take care.
Thank
Bye-bye.
you.
See you later, guys.